Following today's program, we will open the floor to attendees to ask questions. Please utilize the Q&A box to submit your questions throughout the program. And we will do our best to answer as many questions as we can following Dr. Marcus's presentation. As I mentioned, we are recording the program and we'll share it at the conclusion of the program. So with that, I'm excited to introduce today's speaker, again, Dr. Adam Marcus. A little bit about Dr. Adam Marcus. Adam Marcus is Winship 5K research professor in the Department of Hematology and Medical Oncology at Emory University School of Medicine. Dr. Marcus serves as Deputy Director for Winship Cancer Institute of Emory University, leading integration of the research, clinical, and educational components within Winship Cancer Institute of Emory University. He has previously served as Associate Director of Basic Research and Shared Resources, and in 2021 served as Interim Executive Director of Winship Cancer Institute of Emory University. He's got quite a few accolades under his belt. Dr. Marcus also serves as Associate Vice President for Research at Emory University's Woodruff Health Sciences Center, and as Associate Dean for Novel Technology and Research Cores, leading the Emory Integrated Core Facilities and Division of Animal Resources. He provides strategic and operational oversight, as well as scientific direction for the Emory Integrated Core Facilities in partnership with Emory University School of Medicine and Woodruff Health Sciences Center for Leadership. Dr. Marcus is also Scientific Director of Emory Integrated Cell Imaging Core, a jointly managed shared resource of Winship Cancer Institute and Emory University School of Medicine. ICI provides cellular imaging technology services and expertise to support the research initiatives of the Winship Cancer Institute. He's a national leader in understanding how cancer cells invade and metastasize, and how to apply this knowledge in developing new therapeutic technologies. Dr. Marcus is leading the effort to stimulate critical thinking and enthusiasm for sciences in children, kindergarten through 12th grade in the state of Georgia. Along with Teresa Gillespie, PhD, he received a $1.2 million R25 Science Education Partnership Award from the National Institutes of Health in 2016 and in 2022. The grant seeks to increase underrepresented minority participation in STEM with a focus on middle school students. With that, please help me welcome Dr. Adam Marcus.
Great. Thanks for the introduction. I appreciate it. So thanks everyone for being here. This is definitely one of the best parts of my job, is kind of talking about science and talking about the Cancer Center, and a little bit about what we do and how we operate and some things like that, and just kind of shed some more light on Cancer Center research. So I think the title of my talk, which I'll pull up in a second, is related to a little bit of the science, because I think that's super exciting, and then a bit about who we are at the Cancer Center, and then I'll kind of go full circle and kind of come back to the science and maybe kind of speculate a little bit up on what's next for cancer research broadly. So why don't I share my screen, and feel free to drop questions in, and like Nicole said, we'll address everything at the end. So here we go. I'll kind of start that. So I just have a few slides. It's just kind of big picture ideas. And like I said, a little bit on the science. So we are the only comprehensive cancer center in the state of Georgia. We're about one of 55 or something along those lines across the country. And our specific mission is pretty simple. It's to discover cures and inspire hope. And we take this mission very seriously about everything we do. It's really kind of folded into three sort of parts, which is research, which I'll talk a lot about, care, clinical care, and education. So I'll kind of allude to that sort of tripartite mission at the Cancer Center. So we're right on Emory's campus. We're across the street from Eggleston. This is one of our main sites, but I want to talk about a little bit how we're called sort of a matrix cancer center. So we spread across the campus, and then talk a little bit about our healthcare facilities as well that are spread across the state. So I want to start, like I said, with a little bit of science. So what you're looking at is actually a slide of that a pathologist would see, for example, of a biopsy, and a little bit on these are actually lung cancer cells. It's something that we study quite a lot here. We have a large lung cancer research and clinical care team, and this is probably one of the biggest challenges that we have. And I kind of want to relate it back to the disease a little bit. And I'm going to, hopefully you can see my mouse, but kind of this like stretch over here that I'm circling with my mouse. I hope you can see it. And it looks different from the rest of the cells that are served down here. These are actually the cancer cells. And what you can see is in the brown staining here is for like a specific protein in a cancer cell. And every cancer cell looks a little different from each other. Also, they look very different from the normal cells, which are at the bottom and the sides there, that are part of the normal lung. And as a cancer center, we ask questions, why are they all different? How do we treat it? How do we get rid of these cancer cells? How do we not impact the normal cells? Very sort of basic questions, but they kind of guide a little bit about what we do. And I think just as sort of an overall tutorial, there are really two key components that make, and I mean, I should say there's many key components that make cancer cells different, but if you would look in a textbook and say, well, what are the two driving factors that make cancer unique? I'll play a couple of videos for you. These are videos that actually come from my lab. We've been using them for a while, but these are actually breast cancer cells. And if you watch this video, this is sped up over many days, but sped up so you can watch the video in real time. And these are breast cancer cells growing or dividing, or if you remember your high school biology or college biology, is mitosis of cancer cells. And this uncontrolled cell division that we're watching here in a petri dish, so it's not quite the same as what is happening in a patient, but this uncontrolled cell division and this lack of essentially a break to stop the cell division is one of the key properties of cancer cells. And this is obviously one of the targets then for how we have treated cancer in the past. So if you think of chemotherapy, for example, essentially many types of chemotherapy are trying to stop this from happening. And we'll talk a little bit as we move on about how the field is changing quite a lot related to this. And then the other component is this idea of movement. And you may have heard of the terms like malignant or metastasis. That is when cells go on to move and they spread. So for example, if someone is diagnosed with lung cancer or breast cancer, for example, or kidney cancer, for example, does it leave that site? Does it, in other words, metastasize to that site? And this is an example of, these are lung cancer cells moving or migrating. In this bottom left, you can see the cells filling in the gap, and in this top right also, these are cancer cells. So those little lines that are separating all the cells looks kind of like puzzle pieces, more or less. Those are individual cancer cells. You probably have like 200 here and maybe 200 up there. And this is overnight. So this movement of the cells, or again, this metastasis of the cells, ends up being a poor prognosis, ends up driving a poor prognosis or a bad clinical outcome. So one of the biggest challenges we have across the world, across the field, is how do we stop this from happening? Because the reality is, if we could diagnose cancer earlier, if we could spot the cancer earlier, the cure rates will go through the roof. So how do we stop this from happening? How do we detect it early enough through screening, for example? And then, of course, if it has already happened, how do we kill these metastatic, these spread cancer cells in the other organs? And if we could solve that simple question, complex solutions, but if we could solve that, that is one of the clear challenges that we have in the cancer field. So, I'm just kind of trying to lay the groundwork on what cancer is. But if we transition a little bit then, and we'll kind of get back to the science towards the end, if we transition a little bit to what we do as a cancer center and how this all relates, these are sort of our 4 basic aims. And I'm not going to go into details, but I just want to provide some high level information. So aim 1 essentially is transformative cancer research. What I just said to you is, how do we conduct innovative, cutting edge cancer research? And that's not just cells that I showed you. It is related to clinical trials. It is related to cancer prevention. It is related to cancer health equity. It's related to how proteins work together. Anything you could imagine around cancer research is basically within focus of our cancer center. But we want to stay at the cutting edge. We want to stay at the forefront of this. That's our mission as a cancer center. And then, of course, kind of relating back to what I said earlier on, how do we then take those discoveries, and I'm not familiar with the word translate, but how do we translate those discoveries from the lab then to the bedside? And that could be in the form of a first-in-human clinical trial. That could be in the form of a new standard of care for cancer patients. That could be in the form of a new screening mechanism. That could be in the form of increasing HPV vaccination uptake. So there's many ways for us to translate discoveries that are made, we'll say like at the bench, for example, but actually more discoveries could be made even at the computer, right? When we think about like data and data science, how do we then bring that to patients? And how do we also learn from patients and bring that back to the lab? I think, and I'll move into the third aim, and that's really around prevention, right? How do we reduce the burden of cancer? How do we ensure that everyone has access to care? How do we ensure that everyone has access to the most innovative care? And we do this through research. We do this through implementing care facilities around the state. And this is a large part of what we do, and the integration of care and research is a big part of what we do. The last aim is around education, training, and mentorship. So how do we ensure that we have the next generation of scientists? How do we ensure we have the next generation of clinicians? How do we ensure that we're training our staff to be elite, to be top-notch? And do we provide the mentorship? I will say, threaded throughout all these aims, we have several diversity, equity, inclusion initiatives. We have several initiatives that are focused on health equity for all the citizens and people that live in Georgia, and also really throughout the country as well. And I will say we also have some global health initiatives, but I won't have time to really get to them as well. So just some numbers, because I'll throw it at you just so you have some idea. We're quite large. We have what we call our core members. Those are folks that are running like national clinical trials. Those are folks that are funded for their research. Beyond that, if you include all of our caregivers, our membership goes up to about 400 plus folks around Georgia and around Emory. That includes 31 departments. That includes the School of Nursing. That includes the School of Medicine, School of Public Health, Emory College. And that's why we call ourselves a matrix cancer center, because we're not one department, we're not one school, but really we feed in across the university. And we also have several universities that are also members that are like University of Georgia, Georgia Tech, et cetera. We have folks there as well. We have high-level publications, and I will just spend a little bit of time on these two bottom numbers here. This is our clinical trial enrollment. So many of these are first-in-human trials. Many of these are sort of national clinical trials as well. And these are treatments that patients are able to receive before they are approved, with obviously the appropriate safety data in place. And we have one of the most state-of-the-art phase 1 units. So these are early clinical trials to test some of the latest, greatest therapies, to see how safe they are, to ensure that they're effective as well. So we also have the number of patients that we have the data is a little bit older, but from 2020, we had about 7,000 to 8,000 new patients, and these numbers just keep growing over time. So a little bit on our funding, because that's how we sort of measure ourselves compared to some other cancer centers, at least based in the research spectrum. We bring in about $91 million in total funding. This funding is used to support all the research that I spoke to you about earlier on. So I wanted to talk a little bit about just some of our accomplishments along the way. I mentioned chemotherapy earlier on. I will say where the fields are moving to, and we've played a large role in, is immunotherapy. And what really immunotherapy is, rather than just going in and trying to kill the tumor cells with chemotherapy, let's say, what these immunotherapies do, and many of these, almost all of them are approved for a lot of different treatments, we rather enhance our immune system to go in to actually attack the tumor cells. And this could be like directly enhancing the immune system with things like CAR T cell therapy, or this could be with sort of unmasking the tumor cells so then the immune cells could actually go ahead and find them. And we estimate that about 70% of all FDA-approved cancer therapeutics were available in those clinical trials over the past 5 years. So we've played a large role in approving these compounds to then become what we call standard of care for patients. And this is where much of the future is going for cancer treatment. There should be less side effects. It's a completely different approach than chemotherapy, as I mentioned. So this is also a big part of how do we then develop the next biggest treatments. And these are targets that are in our pipeline as an academic university that represent a variety of different targets, and the lines represent what stage they're in. So for example, FDA approval will be the furthest, and we've played a role in several things. And this is FACBC, which is approved for helping diagnose prostate cancer. This is abatacept, which is part of graft versus host disease. And then there's several other targets that may not mean much to you in terms of their names, but essentially these are new targets that have been discovered at Emory where we're developing new treatments around. And many of these will eventually get licensed to pharmaceutical companies to be able to further develop, to be able to further bring out these clinical trials. And this is an important part of what we do is the discovery phase. Actually, many of the compounds that you see that come out from big pharma, many of them have origins in academic universities, whether on the discovery side or understanding the biology behind them. So we kind of see ourselves as generators of these ideas that hopefully will lead to FDA approval. So these are just some examples of that. I did want to mention some things that we contributed to. So I mentioned abatacept for graft versus host disease. We've also played a large role in the lung cancer field for a special type of lung cancer called EGFR mutated lung cancer. This is a compound called osimertinib, we played a large role in the practice-changing clinical trials. There are several new standards of care that developed for smoldering myeloma, for pediatric high-risk Hodgkin's lymphoma, also ways that we treat patients regarding breast cancer, regarding advanced non-small cell lung cancer. And this is just a little bit of the storyline behind abatacept for GVHD. I won't go into the details of it, but essentially it helps reduce the risk of graft versus host disease, which is where essentially the donor tissue, the T cells, the immune cells that are in there start attacking the recipient's body. So this reduces the risk of that, and now it's serving as an effective treatment for these patients. So I want to talk a little bit about clinical care, and probably the most exciting thing that has probably developed over several years is the grand opening of Winship at Emory Midtown. So this is a large state-of-the-art clinical care facility that opened saw its first patient early May, actually. The really new and exciting part about this building is that patients are in care communities. So rather having to navigate multiple sites in a building, right, especially when it's a stressful time, we know how challenging that is. We do our best to bring our caregivers to our patients directly where they could stay in one room. And this has changed sort of the paradigm of how we administer care at Winship. And you can see we have many different sort of operating rooms, linear accelerators. There's an immediate care facility, so that avoids patients having to go to the emergency room, but rather they could seek care right away in our immediate care center. There's also integrative oncology and survivorship pharmacy. And as I mentioned before, everything that we do is integrated with research. So obviously clinical trials is a big part of this as well. So this opened now right in the heart of Midtown, and we think that this will be a game changer for our patients. I also wanted to say we have several hospitals throughout the state, and this just serves as a map. Obviously, you may be familiar with what we have on the Clifton campus, which was the original site that I showed you. We have strong partnerships with Children's and Aflac on the pediatric side. We have a proton therapy center that opened I don't know, maybe 5 years ago. It's hard to keep track of time. Several other hospitals, including the VA, Midtown, I spoke about, Grady, and so forth. So this just shows some of our clinical research sites that we have. I spoke so about clinical care facilities. I want to talk a little bit about research facilities. And I just have a couple more slides and I'll get right back to the science. This is just the growth of our research site. This is a new building that also just opened in 2023. This is 350,000 square feet of research. It'll host about 1,200 biomedical researchers. It's not just cancer, but we have quite a large footprint. This is cardiology, vaccine center, neurology, pediatrics, and we play a large part. And the nice thing about this building is it's very interdisciplinary. So we imagine some really interesting research will come about when you mix cancer researchers with researchers that do vaccines, for example, or around brain health and perhaps glioblastoma and things like that. We also recently renovated additional space over in Clinic B to bring this up to sort of state-of-the-art standards. So we're always growing on the research side too. I did want to briefly mention just a little bit about education and DEI initiatives, and then I'll wrap up. Just to give you an idea of our educational programs that we help coordinate at Winship, it spans from elementary school to our faculty. So, and everything in between, undergraduate, graduate students, postdocs, staff. These are just a variety of programs that we either directly support or they're supported through the National Institutes of Health. And I won't get into all of these, but happy to chat about it offline. And lastly, as I said, we really tried to thread our DEI initiatives throughout all we do. And we see the several pieces to this, one being the infrastructure to do this, two being the pipelines that we have in here, these pipeline programs related to education. Three, we want to make sure that we're ensuring the growth of careers of historically underrepresented groups here at Emory and also recruiting in that manner. And lastly, how do we evaluate this and remain accountable to this? And we see this as everyone's responsibility here within the Cancer Center. So lastly, some of our focus areas that we have going forward over the next 5 years. As I mentioned before, immunotherapy is a big part of this. How does every patient, right, because this is where the disease is going to that I want to speak about in a little bit. How does every patient receive the proper immunotherapy? And how do we sort of remain dynamic and agile and flexible over time to ensure they're always receiving the proper immunotherapy? I mentioned also discovery and development, new treatment, new ideas is a big part of this. Early detection, this is all about prevention and risk reduction. How do we have the right screening programs in place? How do we know what the markers are for screening? How do we have better imaging modalities? We want to make sure that we promote cancer health equity with the goal that everyone has access to state-of-the-art research, state-of-the-art clinical trials, and state-of-the-art care. And then lastly, as we move to an area of essentially big data, data science, and AI, how do we ensure we have the right technological innovations in place, the right infrastructure to ensure that we're meeting these needs, not just at the moment, but thinking 5, 10 years in the future in sort of this rapidly evolving space. So that's where we are going forward. I want to just end with just a couple slides on some of this is my opinions, I guess, on where I think the future of cancer research is going. And I want to just give a little bit of a primer, and then I'll be quiet. I'm happy to take questions on where I think we are right now which is on the left here, and then I'll talk a little bit about where I think we're going in the next 5 years for cancer treatment. So on the left, we now know that not all lung cancers are the same. We now know that not all breast cancers are the same. Just because we categorize them as breast cancer, or just because we categorize them as prostate cancer or brain cancer, for example, or skin, we now know that's a category that we gave to it, right, based upon where it's located. I think as humans, we've actually evolved the ability to create categories for everything. This has actually been well studied, that we like to group things into categories. And we call these sorts of, and as we sort of evolve, we realize actually in cancer, that's really not the case. So we now know that not every lung cancer is the same. And we call this intertumor heterogeneity. That is, lung cancer A may be different than lung cancer B, than lung cancer C, than lung cancer D. And we treat accordingly now. So for example, if there's a mutation in a lung cancer, and let's say 10% of lung cancer patients have this, we treat that in one way. Let's say the other 10% have a different mutation, we treat that another way. We are doing this now, and it results in significantly improved survival. We do the same thing in breast cancer, we do the same thing in skin cancer, for example, and many other tumor types, myeloma, etc. Now, this is where we are now. This is where we've learned a bit in terms of research. And I'm going to bring in this other concept that dives even deeper, and it's the idea of intra-tumor heterogeneity. So within one patient's tumor, right, there are lots of differences. So not every cell, remember I showed you the cells at the beginning and I showed you that image, not every cell, so one cell sits to another, next to another one, they're not the same, right? And that's what's being showed here. For example, you have these cells that are colored pink, you have these cells that are colored bluish purple, and these cells that are colored yellow. And they're not the same. Even though they're within the same patient's lung cancer, it's not identical. And this actually is one of our biggest challenges, because if we end up killing, let's say, the pink and yellow cells, because they have a specific mutation like I described earlier, the blue cells could still survive and they'll grow out. It's kind of like the concept of antibiotic resistance, where if you don't kill all the cells, it could grow back. That's why you stay on your antibiotic for the full course. Same thing sort of happens in the cancer cells, in that many of the cells could grow back. And this is a big challenge for us, because up until the last few years, we really had no way of how to study this. And I want to show you one really cool technology that allows us to sort of deconstruct the tumor cells and be able to separate them out. And I'll show you that on the next slide. So this again is a patient's tumor. And what you have on this left side here, so this large bulk mass, these are all cancer cells. And we are very good at sort of studying this large tumor, for example, and this bulk of the tumor, we call it. However, we also know that, and this is on this right sort of black region that we drew in, this is called the region of interest. We drew this in with the computer, for example. This group onto the bottom right here, these are cells that have budded off the tumor, that have broken away, that have probably going to metastasize, if you remember that term early on. These are tumors that are invading. We have never been able to study this specific group of cells before. So again, we studied the large bulk, but we've never had the ability, the technology to say, well, what is if we just carve out this region here? How is it different than the other region? Because if you just study the large mass, these smaller amount of cells, which are probably like 50 cells you're looking at, they'll get just lost in the crowd. They're sort of like the needle in the haystack. However, it's this needle that sort of drives the metastasis of the disease. So now for the first time, we have a technology that's being implemented around the country at cancer centers to study these small populations of cells that are probably small, yet they're drivers of the disease. So it's called spatial genomics, and I'll just show you a little bit about what my lab's doing in this, and then I'll tell you about how it sort of relates back to the big picture. So what we're able to do is we're able to then basically, as I showed with the previous slide, draw essentially with the computer a region of interest, sort of highlight with this black sort of, digital marker, I guess, surround these cells, we're able to mark this off. And then what the system does, it literally cuts out those few cells. It needs about 50 cells for it to work. And then we take those 50 cells and we find out on like the DNA and the RNA level. So if you think back to like mutations and DNA and things like that, what makes them special? How are they different here? This 007, this 007, how is it different from 010, for example? And then of course, how is 010 different from 03 or some of these other 006, some of these other regions that are drawn? This allows us access for the first time to intratumor heterogeneity in this way, because we're able to say these cells were leaving the primary tumor. What are their mutations? And at some point in the next few years, I would imagine we're going to be able to ask that question on every single cell. So single cell sequencing, single cell genomics in terms of this concept. We could do single cell, but we can't do it like this yet. So we're getting there. This technology at some point, I imagine, will be integrated in patient care, but it has a few years to go. So pretty exciting. The data actually looks like these plots here. So it's called the volcano plot because it kind of sort of looks like a sort of the inside of a volcano. And essentially things that live up here, genes that live up here and here, those are the special ones that probably change in this 007 pack compared to this 010 pack. So these are things that my lab are studying so we can specifically target metastatic cancer cells. So this is totally my opinion. What does cancer care look like in 2030, let's say 7 years from now? Don't quote me on the year, but I think this is where the field is headed. If you kind of combine a lot of big picture ideas, and really this borders around the idea of as tumors change over time, as this intratumor heterogeneity changes over time, how do we change treatment? How do we adapt treatment with the changing tumor in a patient? And it starts kind of with this idea of single cell analysis that I just brought up. We use that information to assess what's going on in that single tumor. And then we have precision treatment for that patient. So we basically obliterate categories and we say your tumor is very specific and we're going to treat accordingly. We're not going to lump this all together, but we're going to have treatment specifically for each patient's cancer, probably related to immunotherapy and combinations with chemotherapy. And then we're going to do something really cool, which I didn't have time to touch upon. We're going to do a blood draw and we're going to say what's new in that cancer in a year, in 2 years, rather than having to biopsy all the time, which is invasive and requires surgery. Blood draws, which are called liquid biopsies, which you'll start to see commercials for if you haven't already. Companies are already developing these. They're in practice. We're going to do a blood draw, a liquid biopsy, and draw blood to say what's changing in the tumor. We're going to be able to image in the appropriate space. And then this is the important part. We're going to then change and we're going to remain agile and change treatment to match with what is developing in that patient's tumor. So you can imagine something that's really dedicated to one person becomes very expensive. And I think the key part to what I want to highlight is how do we make this accessible to everybody? How do we make this accessible not just in Midtown Atlanta, but throughout the country and throughout the world? And I think this is a real challenge. I think this has a cost associated with it. It has a tremendous amount of complexity associated with it. But I think that this is sort of the way forward in how we're going to do this. I can't say it's going to be 5 years or 10 years, but this is what we're moving to. And I think we're going to get even more effective in our treatments. So as I mentioned, the evolution of the treatment aligns with the evolution of the cancer. And I think that's one of the key take-home messages. So I'll stop there. I'll stop the share so it's a little easier to see everything. I see that there are some questions in the Q&A as well. So maybe if Nicole, if that's okay, I could start with those two.
Yeah, sure. Go ahead and feel free to start there. Yes, thank you for those that have already dropped some questions in the Q&A. If you happen to have any more questions, please feel free to drop them using the Q&A function at the bottom of your screen, or you can drop them in the chat box if that is easier for you. But yeah, go ahead and feel free to answer some of the questions that we're already seeing.
Yeah, absolutely. So Great questions. Thanks, Gail. So essentially the questions are, how do user research get translated through regulatory approvals, right? And then how do you implement this? So we have, a whole office focused around clinical trials and clinical research. A tremendous amount of administration goes around this. And I think that this is a big part of what we do to ensure, obviously, the safety of our patients is number one. Absolutely. And then how do we have the right administration in place? And we're actually held to this by a variety of overseeing administrative bodies across the United States, the National Cancer Institute being another one. So we need to ensure that we are following the correct procedure. And I'm just trying to read the question as I go on. Ensure that we're following the correct procedure, and then assure that our patients are aware and that the oncologists are aware of these trials. So how do we ensure that an oncologist in rural Georgia, for example, is aware of the trial that occurs? We have a lot of sort of, quote unquote, marketing and awareness and education efforts around this as well. I think this is a big part, and you really hit the nail on the head, is how do we ensure that, again, everyone has access to this? I'll kind of segue into the second question. There are 55 major cancer centers. How do they coordinate research? Again, we are held to this. This is a big part of what we do. My belief is that for us to be successful, nationally, globally successful, it's going to require coordinated research across the country and across the world. It's not going to be one person, ends up in the lab one afternoon and says, well, I found the cure for cancer. It's too complex. It will never be like that. So ensuring that we're not duplicative, just like you mentioned, or redundant, we need to be asking questions and solving questions that have never been asked before. And I think that's really important for us as humanity. So how do we avoid this? There are lots of national consortiums that exist where we all present our data together. We actually strive to create collaborations. Mainly we focus across the state, so UGA, Georgia Tech, Georgia State, Morehouse, and so forth. But we also have lots of collaborations across the country. And I think you'd be surprised at actually on individual levels how collaborative folks are in general. People want to work together. I think everyone's extremely well-intentioned to do that. All right, I'll move to the next question. A question about tennis. Yeah, so funny question. I've never gotten a question about tennis in one of these before. How does tell us about your college career as a tennis player and how this helps you lead a team at Winship? So I played NCAA college tennis. That's pretty much all I did growing up was play sort of tennis, and college, same thing. How does that help you lead a team at Winship? Yeah, great question. I think the biggest thing, is dealing with multitasking and jumping between things often and actually keeping calm. I think that's actually something that I really owe to being in stressful situations throughout, growing up, right? And kind of teaching you best practices more or less of how to handle stressful situations. But yeah, I think leading a team at Winship, again, I feel like I've spent my whole career here, so I haven't been anywhere else. So I have no comparator, but I will say it's the most collaborative, most well-intentioned folks that I've met. And I think it's actually fairly easy to work with people here and help lead a group. And I don't know, I enjoy coming to work every day, and I enjoy talking with the people that I interact with most every day. So that I'll move on to the next question. And I guess we're okay on time. We have about 15 minutes or so. So how does FACBC differ from PSA? Right. So PSA is a standard biomarker treatment, right? So it could be done in a blood test for prostate cancer. And in general, PSA levels could rise, for example, as the risk for prostate cancer increases. So high PSA would indicate an increased risk. And I should say, I'm not a physician, but I understand the concept here. FACBC is for imaging. So if you get a scan, for example, and more specifically a PET scan, FACBC is taken up by the tumor and is more sensitive than traditional imaging methods. So PSA is going to be a marker, for example, a protein marker, whereas FACBC is going to be like a body scan, for example, much like PET, or you think it's similar to a CAT scan as well. But it actually looks at sort of the metabolism of a cancer cell. So cancer cells have increased metabolism, and that's what FACBC looks at. So it'll give you more of a whole body view, where PSA is just going to give you a level of how much of the protein is there. So I wouldn't say one is better than the other, for example, but they each have different merits to them. So then the next question, any initiatives focused on prevention, early screening, public health? Absolutely, 100%. Public health is one of our top priorities here. So absolutely still critical. So we have lots of prevention programs, both on the research side and then on sort of the implementation side. So for early screening, we're actually growing our lung cancer screening for high-risk individuals. Breast cancer screening is huge for us as well. I'll also add like HPV vaccination. So if you ask, do we have any vaccines for cancer? I'll say absolutely, we have HPV vaccinations. And I was just seeing the data at a meeting I was at. It was just fascinating in that now that people have had HPV vaccinations for, I can't say 15 plus years, you could actually see that cancer risk for cervical cancer is near zero for patients that had it. So I know people are scared to use the word cure, but it gets us more or less towards a cure if you had a vaccination between ages, I'll say 17 and 25. I can't remember the exact cohorts. So if you have it early on, so there's a vaccine that prevents cancer. So getting folks to take this vaccine is critically important. And there are other vaccines that are, on the horizon for other cancers and sort of different contexts. So I think we'll be seeing more and more of cancer vaccines and public health issues surrounding that as well. So lots going on. I'll actually say that cancer vaccines are probably one of the hottest areas developing, and how that relates to immunotherapy that I said earlier on. So lots of cool stuff happening in that space for sure. Yeah, great questions. Happy to take other questions as well.
Yeah, I'll go ahead and ask a few myself, in case folks have any other ones they're stewing on. Again, feel free to drop them in the Q&A box or chat. But kind of leading off of that last question, in your opinion and what you've learned, what are some of the best ways that people can prevent themselves from getting cancer? I mean, I know it's kind of something we hear more and more about every day. And I appreciate you sharing a bit about the fact that vaccines are playing a part in that. But for the average person, what are the best things we can do to protect ourselves?
Yeah, absolutely. So again, I'll preface it with saying I'm not a clinician or a caregiver, but in terms of what's been published and what's out there. So one, if you're deemed high risk, maybe through genetic mutation or increased risk, let's say through smoking, for example, getting screened is probably one of the best methods for prevention. In lung cancer, this could be as simple as an X-ray, or for skin cancer, let's say going to the dermatologist annually, for example, or for breast cancer, being BRCA positive, for example, or getting yearly mammographies, same thing regarding colonoscopies as well. So I think screening is a big part of this. I actually saw a talk a few years ago by the World Health Organization, and they listed several things that say how to dramatically reduce risk. So I'll kind of mention them here. In general, they're things you wouldn't necessarily be surprised to hear, right? Eat your fruits and vegetables, right, over the course of a lifetime. Wear your sunscreen. Avoid smoking, right? And exercise. Essentially, I know they sound simple. You've probably heard these forever, but I think they said that it decreases your risk somewhere of the order of 30 to 50%, depending upon where you live. So those are life changes, right? And those are always the most difficult to do. But those are clear ways. So screening and I guess prevention, I'd put those in those two categories. And then there's always some risk involved, right? It's not 100% thing, right? It's not, if you do this, it will be that way. There are other probably genetics involved that we just don't know yet. What are some other mutations? You probably heard of BRCA mutations in breast cancer, but what are some other mutations that could predict risk? We just don't know yet. And that's where those are the big roles that research play in some of this, because all these sort of mutations that we discover, almost all of them come from university labs and cancer centers like ours. Yeah, great question.
Thank you. Yes, we just had another one pop up in the chat if you'd like to take that.
Yeah. What can be done to prevent recurrence, right? So I think this is a tough question, right? Recurrence is a big issue. I would say following the treatment guidelines of your caregiver, how often it is to get rescreened, how often it is to see your caregiver, your oncologist, if it is a recurrence. There are more and more treatments around or preventing recurrence. I think this is one of the big areas that instead of, let's see what happens, through screening and having your exams, what can we do to actually prevent it from a treatment level? I think a lot of areas of research are on there. So I think, ensuring that you're getting the appropriate screening done, recommended by your caregiver, ensuring that you're seeing your oncologist as well, and ensuring whatever sort of treatments they prescribe to prevent recurrence. Because as I said, there's more and more going on, I think is a big part of this. Yeah.
Thank you. And I'll just have another one or two questions before we wrap up. You spoke a little bit about the NCI designation at Winship, and I'm curious to hear a little bit more about the advantages that one might get from receiving care at an NCI cancer center like Winship.
Yeah, so our NCI designation, I mentioned Comprehensive Cancer Center. So for us to get there, it's a lot about the work to get there. So for us to get that designation, we have to be as innovative, as connected to our community as possible. So for us to achieve that, it's not like we could wake up one day and say, we want NCI designation, right? This takes years. So I think if you then sort of flip that around and you say, what's unique and innovative and what could you get at Winship? It is those first in human clinical trials. It's access to state-of-the-art care. It's access to caregivers that only specialize in usually one specific cancer. So they specialize in lung cancer. They may even specialize in one specific type of lung cancer, for example, or breast cancer, or prostate, or kidney, or brain, for example. So I think that specialty means they've seen thousands of patients and really stay up to date with the latest treatment guidelines. And they provide the latest and cutting edge treatments possible. And behind all that, is the latest research that's trying to be translated to the clinic. I think that to me are some of the biggest advantages. And by no means I'm saying you cannot get great care outside of a cancer center. But I would say we are held to this standard for us to receive designation in 5 years again from now. And we work every day to achieve that.
And how would you say, what role does philanthropy play in Winship's NCI status and the ability to be flexible in its research?
Yeah, I would say almost, I don't know if I want to say most, but maybe almost or most of our ideas of research are essentially start with some form of development or philanthropy. Because for us to get the larger dollar amounts, for us to do high risk research, the federal government usually wants to play a little bit more conservative in what they fund us. Whereas the high risk research that will allow us to then apply for those multi-million-dollar grants that allow us to grow programs in that space require an initial investment, and most of that comes from philanthropy. So I could just speak for myself, and I know this is true through many, almost all the large grants, almost all the ideas, almost all the medicines that we're testing were seeded. Yeah, I think all of them for me were seeded through some form of philanthropy. So I think while it's definitely playing, quote unquote, the long game in that way, that investment gets everything going. It gets everything set up to move things forward. So I think, if you're looking to do something game changing, something different and something innovative that I think all of our philanthropy goes to research, then I think that's sort of one way to get involved. Yeah, those are my thoughts around that.
Great, thank you so much. Well, with that, I hope everyone can join me in giving a huge thank you to Dr. Adam Marcus for being here today and sharing this incredible insight and the work that is happening at Winship Cancer Institute. So again, we are so grateful for you all for taking the time to learn with us. If you would like to support the work that Dr. Marcus and his team are doing at Winship, please consider making a gift to support the Winship Directors Fund by scanning the QR code that you see on your screen or by visiting together.emory.edu/Emory Sparks Hope. This fund in particular provides unrestricted support to areas of greatest promise in the fight against cancer. So again, thank you so much for joining us. Thank you to Dr. Adam Marcus. And with that, we will go ahead and wrap today's program.
Great, thank you, everyone.
Great, and thank you so much, Dr. Marcus. Everyone have a fantastic day, and we will be sharing the recording out after this, so you will have the opportunity to rewatch or share with your community and friends. Thank you.